And I want to just start out with where we are in 2024 with the urobiome, what we think we know. And the biggest thing in about a dozen years since the urobiome was really discovered and replicated and confirmed is that the presence of microbiomes is normal. So this is especially difficult for clinicians, and even more than 10 years into replicated studies of the urobiome, people are still thinking, no, any microbe we get, it's contamination, but that's just not true. It is normal to have living microbes in the human bladder. And it's hard for the clinicians because the clinical test of the urine culture comes back and says no growth, and that gets translated very casually to a sterile urine. And people have an idea, if I give an antibiotic and I kill this UPEC, this uropathogenic E. coli, I will, quote, sterilize the urine. Nothing could be further from the truth. As a matter of fact, absence of microbes, the beneficial microbes, may actually be important. And this is especially important in the women who are affected with recurrent bladder infections, because they have so much antibiotic exposure that we wipe out the bladder beneficial microbes, and we leave them basically defenseless. So we send out the football team with nobody but the quarterback, and there's nobody else on the field, and there's no way they can win that game. And we've also moved past simply counting the microbes. Oh, we have this number of microbes available, blah, blah, blah. We can actually now begin to talk about community characteristics and have an increased appreciation for the various viruses that are present in the bladder and the fungi, and how they act and interact with the bacterial microbes, and the products that those microbes produce and how those modulate community function in health and disease. And this isn't the easiest thing to study in human. We're appreciative of the models and animal models, although those have significant limitations, as we heard from earlier presenters. We also are getting some better insight into health, stability, variability, and we recognize that the bladder is a pretty dynamic community, the urobiome is, and we know it can vary with hormonal status, sexual activity, and menstruation. And just like our prior presenters, the variation that you may see, and I'll show you some data on this, within an individual is greater than, between individuals is greater than what you may see in individuals. So these are the things that we're pretty confident about, and we've made other progress. So I want to recognize and acknowledge my longtime collaborator, Alan Wolf, and I've highlighted his email here. And you may want to jot that down because he has a huge bacterial isolate reference collection that he's happy to share. And this is really important because with urobiome research, many of the microbes weren't known, they weren't indexed, they weren't in reference libraries. And so you would take your sequences and try to match them in a library, and you couldn't find anything. And this has been significantly rectified. And so we not only have isolates that have been grown in cultures or modified cultures, but also the sequences. And this 2024 reference down below catalogs the phylogenetic diversity of the human bladder bacterial isolates. So there's a lot there. And we also have grown these isolates from humans. So hopefully, for those of you who are interested, this isolate collection could be of interest for you. And I did ask Alan if it would be okay to share his email, and he said yes. But I want to turn my attention to an NIH-funded effort from the NIDDK Institute. This is an image of the many, many people who are working on this work. And we don't have pictures of our community partners. Our last speaker talked about the importance of community-engaged researchers, and that is a philosophy that our research group, our research consortium follows. So you can see on the map on the right side, the map of the United States, the red zones are where the clinical research centers are. And up in Minnesota, the green dot is where the scientific coordinating center is. So one of the simple things we started out with, if we want to study bladder health, okay, we know that some microbes are associated, E. coli is associated with UTI. Well, what's in a healthy bladder? Well, there was no definition of bladder health. So we had to do some very stringent fundamental work to just basically begin with definitions. I've put the references here just so you can get a sense of the timeline. So in 2018, we proposed a definition for research purposes just to talk about bladder health. Lots of definitions about symptoms and conditions, but nothing simply talking about health. And once we had that, we developed an instrument to actually measure the spectrum of bladder health. And this was just published in 2023. This is the bladder health scale. There are 10 scales within that. And the bladder function indices, there are 6 indices. And this is a validated measure to measure bladder health all the way from terrible to wonderful. And there's also an adaptive behavior because clinicians know, girlfriends know, that some people adjust their behavior to avoid symptoms or avoid problems. And so if somebody's doing a lot of workarounds and a lot of adjustments, they may say, I don't have any problems, I don't have any symptoms, but they're doing a lot of workaround to keep themselves in that state. So we also have a way to adjust for that kind of adaptive behavior. So the community members gave us the name RISE for Health. RISE doesn't stand for anything, it just is RISE. And it's an ongoing population-based cohort study of adult women designed to inform the factors that promote bladder health and, importantly, prevent lower urinary tract symptoms and then help us estimate the distribution of bladder health. So large study, over 3,000 women. And we have kind of divided the sub-studies into this larger Venn diagram of all the RISE individuals. I don't have time to go into all the study design, but this is a large longitudinal survey study. And within the first year, individuals who completed the 2-part baseline survey were invited to complete an in-person visit. That's the inner circle there. And of those, about 100 were asked to complete a biospecimen study where they collected repeat specimens at home before they came to the clinic visit for their research visit. And I'll share some of the data there. So when we first started urobiome studies, we were very, very careful. We did suprapubic aspirates with a needle control going through the skin so that we were very, very sure that we weren't just measuring skin contaminants, et cetera, et cetera. We used transurethral catheter specimens. But although that's really rigorous scientifically, it's just not pragmatic for large population longitudinal studies. So we began to look at the voided so-called genitourinary specimens, and we used a PZ device. I'll show you that. I don't own any stock, or I don't care if you buy that device, but it reduces the non-urinary microbes by about 10%, give or take. Don't quote me exactly, but it's a different way to collect a voided urine specimen. But then what PLUS did is we looked at the feasibility of getting a specimen from a research participant or a research partner at home versus if that individual comes to the research center and self-collects her urine there, and then we got to ship it to the lab and get it to the lab and all those steps, tremendous amount of work to do that. Does it compare? And basically, yes, I'll show you that data in just a minute. And then we looked at the short-term variability and the concordance from day 1 to day 2 at home, the home specimen to the research center. So we have some of this basic data that can be used to inform future research. So up here in the upper right is the PZ device. This is made in the UK. The funnel is just held up against the perineum and the individual pees. The first about 10 mils collect below, and then the specimen tube fills. And when it fills beyond that, a weighted sponge in the bottom falls out, and the rest of the urine just goes into the toilet. So that's basically how that works. The device doesn't work all the time, and it's best if somebody has, moderately full bladder, at least medium full. So it's not good if someone's only going to give you 10 mils of urine. But we try to figure out, can home specimens be feasible? Because that can open the door for large population research, and we can begin to have specimens sent in from broader populations. And then we have to make sure scientifically, rigorously, is the home collection, is it terribly contaminated, or people dropping their makeup in there, or who knows what else is going on in there? Is it concordant with what we see in a research setting where there's a little bit more control? So we this is the diagram of looking at these, how these specimens compare. Very busy slide here. But what this shows is all the steps that have to happen for someone to be able to send a research specimen in of any quality. And so there has to be a kit that's carefully designed. We put a DNA stabilizer in. We use Assay Assure. Again, I don't own stock in that company, but some DNA stabilizer. We did the benchmarking study to know how long it could be out at certain temperatures before it needed to be in a -80 freezer. We needed to organize the shipping. Turns out you can't take a urine specimen into FedEx and ask them to ship it. You can put it in their drop box, but you can't hand it to a person. These are mysteries that I don't quite understand, but these are the kind of things that you have to troubleshoot. And we had people who put stool in the urine specimen thing and specimen of the urine in the stool thing. And, you know, despite careful instructions that were carefully reviewed by our community members, you just, if it could go wrong, it did go wrong. Maybe not enough volume. So we actually looked at how many we actually got in a sufficient way to the lab. And if you just focus on the circles, you won't have time to read the rest of the slot. The bottom line is about 20% from home and about 20% from the research center don't make it for whatever reason. So if you're going to plan a sample size, you need to inflate it for that 20%. So if you need 100, you got to plan a little bit more than that. So that's the first thing we figured out. We hope that helps other researchers as they begin to do population studies. The concordance part is good. The scientific portion was solid, and we can now say with reasonable scientific confidence, supported by data, is that participants can self-collect urine at home, not in any way the way we did it in this study. And there's a sufficient short-term similarity and concordance between the home sample and the research sample. We didn't see a lot of variation within individual research partners on this. So we feel fairly confident about that. I'm going to turn away. We are having some early data that's being run on the actual status of bladder health, but I'm not quite ready to talk about that tonight. We need about 2 more weeks. I'm sorry it wasn't ready for this presentation, but it will be soon. And we'll make all these sequences from these microbiome studies available. And they're all being put in the urobiome bioinformatic, forgot the exact name of it, but they'll all be publicly available, and the NIDDK repository will have all of our data as well with these urine sample sequences. I just want to highlight some other longitudinal work. We talked before about looking at longitudinal samples, and this is a study of 8 premenopausal women. Keep in mind the urobiome of premenopausal women is distinctly different from that of postmenopausal women. These women did daily assessments over 8 months. They were embedded in a larger study looking at a probiotic, which didn't make a difference. And so we're able to pool them for this analysis. And so this first report came out in 2020, but I want to show you this. These are, for the most part, unique individuals, except individual 7, which is in the third panel. G and H are the same individual. And just like you saw in the other microbial niches, you can see that there's variation day to day, but by pattern recognition, these folks look pretty different from one another. And we could see rapid shifts. And in fact, we could tell when people were getting when they were about to get a UTI about 5 days before they got a UTI. Now that's not clinically actionable. You can't just collect urine all day just to check. But you can just see that people varied significantly. And these are all healthy people without symptoms, without vaginal symptoms, without urinary symptoms. So one of the other big take-home points is you can have any number of a wide range of microbial community states and have a healthy state, or at least an asymptomatic state. There's so much more to learn on this, though. Going beyond just the microbes, the other aspect is the metabolomics. Now, we know urine has a lot of metabolites, but we don't know much about the relationship between the microbiome and the metabolites. And one of our early career faculty at UCSD, Lindsay Burnett, is leading this work. And we've just finished some of the benchmarking studies. We weren't sure that we would be able to do it, metabolomic studies in the presence of urine that had been stored with Assay Assure. And when you do the metabolomic studies, you can see the assay is sure it's a distinct chemical signal, but you can just set that aside, and the rest of the metabolomic studies are working fine. So she will be running metabolomic studies on all these samples from the NIDDK study. So we'll have about 500 individuals. Well, we maybe not do all of those people, but at least a good portion, several hundred people will have both their microbiome and the metabolomic signals for those communities. I want to just make a quick comment about the mobile genetic elements of phage and plasmids. I do think this will be one of the new frontiers. We think about plasmids carrying bacterial resistance for antibiotics, for example, but we think that there may also be some signal that plasmids affect permissivity in phage therapy, and phage biologists are probably all over this, but there's an intersection between the microbiome and these mobile genetic elements. And so we may be able to, as we move into phage therapy for recurrent UTI, we may be needing to understand this a little bit more. And this has not been well characterized yet. We know there are distinct phages in the urine system, and we know that some plasmids can carry the resistance genes. And so we'll be looking for a little bit more about this in the next phase of our research. So there's so much more. We've opened so many doors. The research community worldwide has opened a lot of doors in urobiome research. So we really need to understand the microbial signatures that are associated with health, what the metabolomic signatures are, where the urobiome microbes are coming from. Everyone just assumes it's the vagina or the gut. Maybe, maybe not, maybe in different people or different circumstances. Metabolomic studies may be able to help us because they've got some pretty cool bioinformatic systems in metabolomic world. And we need many more studies of temporal stability and resilience in diverse populations and in diverse clinical situations. We need to understand the dynamic and intricate interplay with the host immunity and the host health status, and a correlation with host genetics. We really don't have much of that at all. And we need to understand how these communities function. What are the interactions between the bacterial, virome, and microbiome? We really don't have a good sense of that yet. And we have essentially no information, very, very few studies on the health and the concordance of microbiome and the urobiome and other sites nearby and other sites elsewhere. Most importantly, we really need to be able to find out, are there clinically helpful therapeutic actions for preserving a healthy urobiome or restoring one that's been dysbiotic or been disrupted, helping it recover? Are there ways that when we give antibiotics for UTI, there's something that we should do to accelerate or help with restoration of a healthy urobiome? We just don't know how to do that yet, but that I think will be able to be determined over the next couple of years with the dedicated researchers we have. So thank you for your attention today. I just want to highlight the urobiome consensus paper. One of the reasons our urobiome research has been able to go quickly is that we've been able to work together and share data, share our methods clearly and cleanly, and that has been facilitated also by the STORM's reporting guidelines. And so I encourage everyone who's working in this area to adhere, and I want to highlight Dr. Lisa Carson's work in both of these documents. So I just want to close with, on behalf of the patients and research partners that we work, yes, there's much more that we should know, and we look forward to learning a whole bunch more together. Thank you for your attention, and I look forward to your panel, the questions during the panel discussion.